Good morning, everyone. I'm Choi from UCLA. Today, I will talk about our multi-omics data sets acquired from mouse model to human cardiac disease. And then I will briefly talk about the machine learning based strategy that capable to integrate different omes, basically by finding the hidden association between proteins and metabolites. So thus, it may provide a better understanding of molecular mechanism of cardiac remodeling. As a dissemination mechanism, the strategy being studied and established in this project, deployed in the MetProt, which you may heard about before in my previous presentation. As a brief recap, MetProt is the computational platform in the cloud that help users to analyze and integrate their multi-omics data sets, especially in metabolomics and proteomics. So my previous presentation about the MetProt can be found in the YouTube link here. So here's a brief outline of the presentation. I will start by giving background of the heart failure as a significant public health issue. And I will also talk about the relevance of plasma metabolomics and cardiac proteomics in clinical heart failure and current challenges of multi-omics integration. and then I will talk about the aims of study and multi-omics data sets, including the experimental design, and I will study the reliability of the multi-omics data sets. And then I will talk about the methods of machine learning based strategy for integration and quick preliminary results. And I will quickly show the demo at the end. So I will start with giving some background and the motivation of our study. So why study mouse model to the human cardiac disease? So heart failure is the leading cause of death in US. According to the AHA statistical updates, there is more than 6.6 million heart failure patients in US, and approximately 670,000 new cases are reported every year. And among the patients, 80% of them are hospitalized, and 50% of diagnosed patients died within 5 years. So why proteins and metabolites are important? So proteins is an end product of the central dogma, and the metabolite is an intermediate as well as an endpoint product of the metabolism. And more importantly, the proteins and metabolites are essential building blocks that collaboratively formulate biological pathways. So thus the phenotypes are most closely reflected by the dynamics of both metabolites and proteins. So the largest scale multi-omics approach may provide the mechanistic insights of the complex pathophysiology and cardiac remodeling in failure, as well as it may assist biomarker discovery to support patient stratification and clinical classification. Despite the importance, proteomics and metabolomics data sets are often fragmented and rarely integrated. More crucially, there is a lack of computational methods and technical platform for integration. In the Aztec Discovery Index, we found approximately 1,000 proteomics tools and databases, while we only found 32 metabolomics application databases. And to our best knowledge, the availability of application that somehow comprehensively analyze and integrate both omics is currently very limited. So this limitation hinder our capability to actually identify driver molecules relevant to the certain disease. So in today's talk, we have 3 specific aims. So aim 1 is to study the reliability of our benchmark multi-omics datasets obtained from 6 genetic mouse strains. And aim 2 is to establish machine learning-based strategy for proteome and metabolome integration. And lastly, aim 3 is to incorporate the method into the MetProt and also develop the visualization tool. So first, I would like to introduce our data set. So when the mouse is under cardiac stress, it develops hypertrophy. And to do so, we injected isoproterenol through the osmotic pump and up to 14 days. And we collected cardiac and blood sample 1 day before the treatment and 1, 3, 5, 7, and 14 days after the treatment. So, and then we collected the cardiac tissue. For the cardiac proteome, I use the reference data set produced by Edward Lau and other colleagues in my lab. And we have profiled protein abundance in 3 subcellular fractions, including nucleus, cytosol, and mitochondria for the 7 time points and 2 different conditions, which are the ISO and control. Also, we profiled the protein half-life for the 3 subcellular fraction as well, and in 2 different conditions, same ISO and control. Also, we have profiled the plasma metabolites in 6 time points in 2 conditions, and we repeated the profiling 3 times. So we have 3 technical replicates. In addition to that, we have extracted the SIMS information from Jackson lab. Also, we measured the heart rate body rate ratio, which is a phenotypic characteristics of hypertrophy. So for the protein abundance data sets, we have profiled ranging from around 6,000 to 6,600 proteins in 6 strains for the 7 time points and 2 conditions. And for the protein half-life data set, we profiled proteins ranging from about 1,500 to 2,500 proteins and for the 6 strains and 2 conditions. And lastly, for the plasma metabolite concentration, we identified and quantified 595 metabolites for 6 strains and 2 conditions and 3 technical replicates. And in total, we have 1,663,932 data points. So according to the Wikipedia, the isoprenoral activate the beta-1 receptors that induce the positive iotronic and chronotronic output of the cardiac muscle, and leading to increased heart rate and blood pressure. Also, it activates the beta-2 receptor, which induces smooth muscle relaxation in the lung, and gastrointestinal tract, uterus, and various blood vessels. Increased heart rate and heart muscle contraction is also associated with the beta-2 receptor. So this is the so the question is, can this data set explain any known phenotypic characteristics? So we have performed the differential expression analysis on the protein half-life data sets, and we were able to identify 62 proteins in cytosol, 38 proteins in mitochondria, and 39 proteins in nucleus that significantly changed in iso group over the control group. And then we performed the pathway enrichment analysis on the significant proteins in each organelles. And interestingly, muscle contraction was commonly detected as most significant overrepresented pathway in all three organelles. And also we performed the differential expression analysis on the plasma metabolomics data sets, and we were able to identify 58 metabolites significantly altered after the isotreatment. And interestingly, aldosterone is overexpressed in the plasma, and there's lots of evidences that show that the aldosterone activation increases the ROS, and the ROS regulates calcium transportation in cardiomyocyte. And since the calcium transportation is important for the cardiomyocyte contraction, it increased the cardiac hypertrophy. So those evidences actually indicates that the data set we have collected are sensitive enough us to detect significant signals in cardiac remodeling. So we have established a machine learning based algorithm for proteomics and metabolomics integration. So the study object is to identify the subset key feature, basically which are the signature protein and metabolites with given discrimination capability. So in approach, we use a random forest recursive feature elimination algorithm on the protein and metabolite combined data set. So basically, this algorithm randomly select the subset of features and then create a model with the chosen subset and calculate the distance between two groups. In our case, is ISO and control, and eliminate the subset with a low discrimination capability, and then recursively doing it over and over until get the best combination of the features with a maximum discrimination power. So we use training data sets for the feature. We use a 535 molecular values, including protein abundance value and metabolic concentration value that that shared in both ISO and control group. We have 72 observations. That is 6 mouse trains, 2 treatment groups, and 6 time points. And we have 2 labels, which is ISO and control. So this slide illustrates the signature molecule extraction results for iteration number 9. And it identified 26 key features, including 24 proteins and 2 amino acids. And it was able to yield very high accuracy, about a 96% accuracy. And as you can see in the 3D plot, PCA plot on the left side, you can appreciate the clear distinction of ISO from the control group. Actually, this is end of my presentation, and I can quickly go over the demo. So we are still under the development of the random forest-based feature extraction, under development of the extraction method, but we already implemented the visualization tools. So you are going to have 2 different types of plots. One is
you don't run HSP, really? Yeah, I mean, it's probably not in yet, but you could call them. I mean, I could probably help you out. I don't know what's going on.
So one is the static plot. You'll be able to download as a high definition.
It'll be a little while. So
also, you can have a dynamic plot that you can see different angles.
Okay.
So as I said, the feature extraction method is still under development, but it will be incorporated by end of this month, and the visualization is already implemented. So that's the end of my talk, and thank you for attending, and I'm happy to take any questions. Thank you.
Any questions for Howard?
Hello, I do have a question. How do you combine the proteomic and the metabolic together? So did you simply put the 2 data metrics together?
So what I did was the data range, the data range are different.
Mm-hmm.
So what I did was I basically use a fold change of ISO versus control. So all the data that used in the matrix is basically the log to fold transformated fold change.
Okay. So you actually use the fold change data and put them together.
Yes, yes.
Okay.
To make things in make proteomics value and metabolomics value in the same range.
Why don't you normalize these two data sets in like you normalize within data set and then do global normalization and put them together to do the feature selection?
Yeah, definitely that normalization will put proteomics data and metabolomics data in the same range. So definitely I will consider, I can consider, I think it totally makes sense using the normalization as well, but we thought the log fold change may contain more valuable values. That was the reason why we use it.